So we've got Martin Chutelek from Photon Systems International. These are one of the sort of leading plant phenotyping companies globally, I would say. Martin did his original degree at the Technical University of Brno, finishing in the 90s. He's the owner and CEO of Photon Systems International, which was one of the first companies to offer chlorophyll fluorescence imaging. They're still very active in research and development around photosynthesis and chlorophyll imaging, and as we're here today, many other aspects of plant phenotyping, and they continue to take part in European projects such as the ADAPT project, which some of us here at the Institute are involved in. In 2016, he was the Ernst & Young Tech Businessman of the Year, but still, as I say, maintains an active sort of academic research profile. So Martin, I'll hand over to you.
Yeah, thank you very much. Thank you very much for the nice introduction. So now, today I would like to talk about the future technologies, new advanced technology, which you may use when you do the research and you want to get the Nobel Prize. So when you want to get some when you want to do something special. So basically, I will talk about the neutron, Raman spectroscopy, Raman imaging, and X-ray. So let's a little bit background about the PSI.
What's going on? We'll take a look.
PSI is the company which is doing development of scientific equipment, but we are so weird company because we also do science. So we are developing methodology, and this is why we are so close to the application and understand what are the needs when you do the research. When you want to invent something special, you need instrumentation. If you want to go to another galaxy, you need a spaceship. Right now, there's the Webb telescope and satellites, and with this super special equipment, then you may get new knowledge and new information. So again, when you do the research, you need tools. You need special, unique tools, to be able to more understand, so what is happening inside, to more understand and the basic research. We are one of our focus is not only that, but one of our focus we are developing equipments in terms of the plant phenotyping, and we have focus on the state of our technology. So we are trying to insert to our system as much different techniques to see the problem from the different perspective, so we can then go really deeper, and combining this information together, we may better understand what is happening inside. Because right now it's not only that if you have one signal, I should say, one type of instruments, and the information is covered in the noise. But if you are able to put more instrumentation, so you may again looks from the different perspective, you may combine this information, like everything is covered in the noise. But if you will put this together, so then you may get the information which is hidden inside. And the phenotyping, I think all of us, all of you are quite familiar with what means the phenotyping. Um, so you what we are doing, the systems when you have your plants in some growth area, when they grow, where you can apply different conditions, and then you are moving these plants through the imaging stations, so through the sensors, and then you may pick up the information from the plants, and then they again return back to the area where they are under the specific condition. And what you may do, you may apply different type of stress, biotic stress, abiotic stress, again to see how they react. But you cannot see this by eye. You just not able to recognize so what is happening by eye. So you have to use the equipment, and you may look inside, to understand the physiology, to understand and morphology. So how the plants react on the biotic stress or abiotic stress, or how they react. You may also do really basic study, so you can understand the functionality of the genes, using such kind of the system. There's a number of sensors which we are at the moment using, but I will focus on the three type of the sensors, which they are quite special, and it's X-ray, neutron imaging, and Raman spectroscopy. So there's three main guy. They are responsible for the development of the three different techniques. And as I said before, if you are able to collect this information, so you may really put all this information together. So you may have this like piece of cake of each part of the system, like to better understand what is happening, generally in the system. So first of all, okay, so what is the differences between X-ray and neutron imaging? As you can see on the picture, X-ray, it's more absorbed by the layers, the material, and doesn't penetrate really deeply inside. So if you are using X-ray sensor, if you go to the airport and you will have gun covered in your suitcase, then if you will put some metal plate, so you may hide the rest, so the gun, because everything is absorbed by this metal, heavy metal plate. Neutrons are different, so they are able to penetrate, to go through. So then you may deeply, see what is really inside. The absorption is different. On the other hand, of course, there is some effects which some of the materials scatter them, and you are not able to get the enough contrast. So this is the basic difference between these two different principle, X-ray or neutrons. Neutron imaging is it's very interesting things, but on the other hand, it's very difficult to have it in the lab, because what you need the source of the neutrons. Usually it's nuclear power station, so you have the reactor for this. And then this reactor will generate the beam of the neutrons, so you have the beam of the neutron, and you can use it to image the sample, and how neutron reacts with sample which is inside the beam of the neutrons. What we are doing now, we did a lot of experiments now in Prague, where they have three nuclear stations, so three reactors, different power. But the main our interest was to use just the regular source when you may have some material which is generating is radioactive, but on the other hand is generating the neutrons. So you may have this material, and then you may have the small small source of neutrons, and then you can use it in the lab. And we were developing together with some British company the detector for that application. And this is the one example of the neutron imaging. Once again, neutrons are non-destructive. If you have, and so what we have at the moment, we have the camera, which is able to detect individual neutrons. So we need very low radiation, low intensity, and using that, then we will not affect, so we will not damage the plant. So this is why I'm saying it's non-destructive imaging. And we can then do the scan, so we can see inside to the object. One of the application, because the neutrons are really scattered by the water, and this is the example when you can see the water distribution in the soil. So how water, penetrate, and then how is distribute in the soil if you will apply the soil in quite fast time window. So you see, this 300 seconds range when we can do the imaging, and we can detect this water distribution in the soil, and of course also in the roots. This is the one thing which is, when I want to compare with the X-ray, it's with the X-ray, it's very difficult. There are the people which they are doing root imaging using the X-ray, but it's very difficult because there's small differences between the absorption of the soil and the roots. So it's very high, very difficult to get the high contrast between individual roots and the soil, especially if you water soil. Another another application when you can do the the imaging about the water uptake. We were focusing at the first stage, something which will give us quite good contrast, and it's not super difficult. And the water is the good example. So everything about the water distribution and the plants is the one application for the neutron imaging, because the water is really scattered and absorb the neutrons. So you can do that. Our sensor is also allow to use the different type of neutrons with the different energy, which also works as the hyperspec. So you may use the different type of the neutrons, to select the optimal to be really optimal for the specific application. The second part is X-ray imaging. So what we develop and what we are doing at the moment, so we mount a sensor and the source of radiation on the robotic arms. So then you can do 3D tomography. So you can scan everything what you want. You may select a specific area and then just focus on the scan. You can do the 3D reconstruction. So we add one more camera, just RGB camera, before the scan. So we are taking the picture. We do artificial intelligence to recognize the object of the interest. So you may define, okay, I would like to scan this apple, I would like to scan this flower, or the second leaf, in the structure. And then this information goes to the robots, and the robots may just focus point the area of the interest, and they can scan the specific area. Because also, it's about the X-rays. It's about the time. So it takes, you know, a lot of time to get, you know, precision, for example, 3D image, and you would like to minimize the amount of the of the energy which you will put to the sample. On the other hand, also you would like to do high throughput screening, so you would like to minimize the, the time which is necessary for the individual scan. There's the sorry, I would like to change the okay sound. Okay, so this is the example. I hope that you can see the video. Rob, can you see the video?
Yeah, we can see it, Martin. Thanks.
Super. So this is how the system looks like. And the beauty also for this system is that you can mount on these robotic arms any the type of the sensors. So you can then point you know this other sensors to the specific, once again, to the specific area uh, what you would like to use. And this is very important for our Raman spectroscopy, because uh, the Raman spectroscopy is extremely sensitive, and you have to put your sensor so much closer to the sample. And if you want to do the imaging, you just have to follow the object. Right now, I would like to show you some results. You know, some pictures. So X-ray, once again, it goes. You know, if you go to the detector, you can see your bone. The same thing it's in with the plants, but of course there should be the contrast in absorption between the part what you would like to see and then and the rest. So there should be somehow the contrast. The scanner what we are using, it's able to do hyperspec X-ray imaging, which means that we may also record the energy for the individual spectra and split the energy to the at the moment like 12 different channels. So this allow us to get a higher contrast, because we may select the specific energy which is better absorbed or by the object which we would like to see inside. So what we did, it's the okay, we were working with the seeds, so we were looking into the seeds inside. We were yeah, so the seed quality analysis, spike development analysis. So we were doing the record on the time sequence of the spike development uh, from the corn, and one of the big projects, and Rob is also involved with this, is the tuber in the potatoes. So we can see, you know, the development of the tuber in the soil directly. It's the structure analysis. Yeah, in more detail, the seed analysis. So you can really see the morphology, the structure inside of the seeds. This is the comparison between the RGB image, you know, showing 2 years without obvious defects. So you see, you know, there's like by eye, you cannot see any defects. But if you will use X-ray, you know, so you can see that the sum of grains in the spikes are empty. So they are not inside. But you cannot see. So what do we do? We develop now artificial intelligence tool when we can count, you know, number of grains from the sample, and then, you know, use it as the information for the phenotyping system. So once again, so you may, you know, use the different type of the phenotypes, and you may search, you know, how they react to the different type of stress. What is important right now is like the older planet, the better. weather condition. Because of climate change is the different. So yeah, there's a lot of flood in Pakistan, and dry season in the Europe. So all of us know that now is not the time to develop a crop which will produce or will generate a higher yield, because the yield is good, but the crop which will be more resist against the different type of the stress. And this is what you can scan and screen, in our system. And I should say nice pictures. We also have the project with the people from Australia, from NU University, and when we are study early stage of spikelet development. So you can see, before this is visible, you can see, how this part is developed. And that's is important enough to compare, different mutants, and to understand the basic principle. And not the last, but another project again, adapt, when we can scan soil with the potatoes, and we may see the development of the individual tubers, and we can also, like to study the root structure of this, which is not, as I said before, which is not easy, because the differences between the absorption of X-ray, uh, between the roots and soil, especially if the soil is water, because again, X-ray is also really absorbed by the water. It's small, so you have to really, adjust these things, and it works on the edge. But what helps us once again is, because we are able to detect the energy, so we can tune the energy to get the higher contrast of the picture we want to see. And the last picture is, we can easily see, and I'm sure that this is difficult for you, but you can see here bugs, worms inside the wood. And we have another project with Oak Ridge National Lab, where when we do some research with willows. So it's another type of the application. And one more image, when you can count, the seeds easily. So yeah, that's one of the application. Okay, so let's skip these wonderful pictures, and let's talk about more about the Raman spectroscopy. So Raman spectroscopy is it's a very powerful tool to measure metabolites, and especially complicated complex. When you do hyperspectral imaging, so I mean the visible light, then you may you may detect the content of the different pigments, xanthophyll, chlorophyll content, but the structures are quite small, in terms of the length of the chain. So this is the hyperspectral imaging is good for the pigment quantification, pigment detection, but it's not possible to use for very complex systems. And you also know that you may have between the different types of metabolites, you may have very small changes in the chain. And the Raman spectroscopy is focusing on or measuring the vibration of the molecules. So this vibration of the molecules, uh, changing the scattering of the laser signal, and you can detect this vibration, and that means you can get the spectrum, and you may then get the amount of, or quantify the amount of the specific metabolites. Uh, The signal, so this is the advantage. On the other hand, there's of course some disadvantage. The signal is extremely low. So you need complete darkness. You need very sensitive instruments, spectrometer and a specific laser, to be able to detect this kind of the signal. So what then? This is there's some examples for the specific applications. So we were measuring anthocyanins and carotenoids, for example, and we you may also apply the stress, as I said before, light stress, drought stress, cold stress to the plants, and you can see non-invasively with the Raman spectroscopy how the content of anthocyanins and unconsciousness were changed during because of this type of stress. So you may see the effect. So what is happening inside of the plants and what how they react in terms of metabolites to the stress. What you have to develop, because at the moment it's like you have such kind of a spectrum and you have to develop the database. So what we also doing, we are using standard classic method like HPLC, you know to do the extraction, to measure the amount of anthocyanins, and then to correlate, to develop the database when you can correlate this information with the information which you will get from the Raman. And then you have such kind of so you can link the individual spikes, individual peaks from the spectrum to the information to your knowledge you have because of the standard instrumentation. This is the example how the spectrum looks like. Uh, as I said, so you can see the chlorophyll, the individual peaks from the individual compounds, and what we were doing, we were linking, we were calibrating the system using the standard HPLC analysis. Another type of the spectrum, so another information. Sometimes you are getting the number of the spikes and nobody understand what is this. But you have to what you have to do. You have to link this individual speaks together with the individual metabolites. The one problem which you face with the Raman spectroscopy is the fluorescence signal, because some of these molecules they generate a lot of fluorescence signal, which then they covered the information from the Raman. So you have to what you can do, you may select, and at the moment we are with like 3 different type of the excitation lights. So you may have select the different light which will be not absorbed by the fluorescent performance, or I mean the fluorescent molecules. So you select the excitation light, where there is the minimum absorption, or let's say no absorption by the fluorescent dye, like the chlorophyll, for example. And then you may do the spectra using this type of the wavelength to avoid the signal, the fluorescent signal from the molecules which are in the sample, and which may completely cover the area and the rest of the metabolites what you would like to measure. What we did here, so we mount to our X-ray to this robotics arms the fiber optics, which will deliver the excitation light and also will detect, so the light which go back to the fiber go to the spectrometer, so it's also used for the detection. And the signal, as I said before, it's very low, so you have to really point, you have to be super close to the object, and to get enough information, enough signal, and to have enough sensitivity. But then the R may follow. So what we may do, we may do the analysis from the individual parts of the object and to see the heterogeneity. So then we may have such kind of the imaging. So this is in principle, this Raman spectroscopy, it's non-imaging because it's collecting just the average signal which is close to the fiber optic. On the other hand, if you spot individual parts of the plan, you may get information from the individual individual part of the object. Yeah, uh, that's all. So it would be great to have some questions.
Thank you, Martin. That was a really interesting and cutting edge discussion of sort of cutting edge technologies for plant imaging and analysis. So we've got our first questions just popped into the chat. Ali, do you want to ask that question directly, or do you want me to just read it out for you? Yeah, you're muted, Ali.
If you can speak me. Oh, I don't think I'm muted.
Yeah, I've got you now.
Hi, Martin. Thanks for that. I'm sorry I missed the very start of your talk today, but re the Raman, how well developed in your experience are the databases that your system would be accessing to allow us to identify unknown organics? Because I know that for some of the industries like the silicon wafer folk, they have brilliant databases and they can identify pretty much anything. But I'm not aware of that being the case for general biology at the moment. And I wonder, would we need a specialist? Would it still require a chemist to be making the most out of that technique? And follow-up question is, how difficult do you think it will be for us in plants, given that there's fluorescence from everything, from all the tissues?
Okay. Okay. So yeah, what is true is the Raman is mostly used for organic, not for non-organic compounds. But in principle, there are no limits. The problem is, again, if you measure the vibration of the molecules, so you need like more complex structure. And what at the moment is not really like done, and this is what we do, is that you really need, with the specific sample, you really need to develop the such kind of the database when you can link this individual peaks, because like it's mass spec, so you are getting number of the peaks and nobody knows what these peaks means, because some of these peaks are just the parts of the another molecules. So because of the method, you have, for example, CO2, so then you have the peak from the carbon, then the oxygen, and individual oxygen, or oxygen 2, and the CO2. So that's like number of peaks, and then you have to link with this individual parts. And the same is with the Raman. The another complication, as I said before, it's the signal which comes. So Raman is not fluorescence. I mean, the classic fluorescence. And if there are some components which generate the fluorescence signal, so they cover that part of the area. And what we do again, we are then selecting another excitation laser, different wavelengths, to avoid this fluorescence. If there is some important peak from the individual components, we were now working, but there is the one new project with the guys from Lathrop University in Australia, and there's the department from defense, which they would like to detect chemical weapons, and the residuum, and one Raman spectroscopy is a very good candidate, because somehow it's if you have, there's on the market some Raman spectrometers, and there you may split to the scientific, extra sensitive, when you can really reach very high sensitivity, and there are some others, like for some basic, I mean the analysis, when you have some very strong signal from the individual components and you just want to detect this. So this is the differences. You need very sensitive spectrometer, to be able to detect small amount of the individual compounds. So I hope, so the unorganic in principle possible, but depends on what are which type of molecules you are talking about, and how they uh, uh, what are the the signals which they provide using this Raman excitation light. If you can really be able to detect and get the good signal to compare what is in the rest in the sample what you have. The second thing was about the imaging, right? About how you can do the distribution of the individual metabolites in the individual plants. Yeah, this is something which, okay, what we do now, we are more working on the macro scale, but we have we publish a number of papers when we are measuring this from the individual cells. But this was the system, mount on the microscope, so you can find our publication. So it's just and you point the different area, and you do the analysis. The problem is some of the signal again is strong, and you may need, let's say, second to acquire enough signal. But some of the signal is very low, and you need minutes. You need the minutes. And then is also proportional between how much exciting light, and if you will not generate any damage, any bleaching, to the sample. So yeah, the question, to answer, it's not completely easy because it's specific for the individual ones. But you can do the imaging for sure, and this is what we did on the macro or micro scale. But I was more now talking about the macro scale, like comparing individual metabolites between the leaves, and different areas in the plants, which works well. That's no, it's like more easy because I can also collect, bigger spot. I may collect, I'm not taking the information from the individual cell, but I am taking the information from the large, I should say, and I mean 1 millimeter, area square, but what large area, which is also, so much helpful.
Petra, you had a follow-on question. Has that been answered, or would you like to go ahead and ask?
Yeah, no, I was just wondering about the sort of the purity of spectra in terms of the potential effects of complex environments and biological conditions. So when you're trying to set up reference spectra and things like that, how does that work?
You mean Raman spectroscopy now?
Yeah.
Okay. Yeah. So Raman, what we have, so we have complete darkness in the system. So because the signal is very low, so you have to avoid any light and the background light. Then yes, the calibration. As usual, it's biology. So it's better to do the comparison. So which really works well, because if you have plants, which they are stressed, and to compare to the control, so you can easily compare the differences. So you can see the differences. If you are focusing on the absolute value, then what we always did, we did this chemical analysis to calibrate and understand what means this amplitude, in the signal. Because yeah, there's a number of FX structure, morphology of the system and blah blah blah. So but for the comparison, it's such easy because so you have the same conditions when you do the measurements, and you can do easily the comparison, if it's 2 times more or whatever.
Okay, does that answer your question, Petra? Okay, there's a follow-on question. We'll stick with the Raman for now. Derek, you had a question, I believe.
Yeah, hi Martin. I've done loads of Raman years ago. Raman was always kind of treated as mid-infrared's evil twin because no one ever used it. So were you talking about FT Raman? So which would be really useful in a biological sense to iron out noise, but also taking it a step further with increases in computing systems. Is the resolution there to create wavelength-based maps, constructed maps of the plant, just based on signals? So, I mean, you could create essentially a lignin map of the plant using Raman, which would be really interesting. What's your thoughts?
No, yeah, I think so. It's one of our main interests, and as we have this number of publications, like we changed the wavelength of excitation laser not to be absorbed. So we went to the infrared. So which that means, because chlorophyll and all these organisms, they are very light sensitive. So they have this Harvard complex. And if you are using classic, green excitation light, so it's or red, it generates a lot of troubles. But we moved to 1000. There's some publication showing we moved to 1050 nanometers, which is the regular wavelengths for communication. So you can easily find the source laser source for that, and there's no absorption, but you can detect the signal. And I should say it works well, because what you can see, there's a little bit absorption in the water, so you may a little bit heat the sample because of but from my perspective, it works well. So this is it's also, it would be great like to have multiple excitation lights, and again, you can do such kind of a Raman hyperspec. But this is a bit complicated.
Cheers, thanks.
Okay, we've got a hand raised in the audience. Archie, I believe you've got a question.
That's better. Hopefully you can hear me. Thank you very much. Martin, thanks for your presentation. You mentioned, forgive me, I'm not a non-scientist here, so my clumsy question, I sort of plead mitigation for. You mentioned its ability to perhaps detect chemical weapons or chemicals being used in that sense. And I wonder if the same would be true for let's say synthetic biological agents that could be embedded in perhaps a tolerant or host, and then introduced in order to cause, which would then cause problems in survive in perennial plants and cause massive damage to annuals and the like, or biennials. And I wonder whether you've any thoughts on that. It's topical for me as I'm advising on another level on food defense matters, and I just wonder whether there's anything in what you've just described that might be relevant. Thank you.
Oh, okay, so yeah, we did some experiments. We were also involved in a food safety application. So I have to say the following. It's always a proportion, how strong, signal you have, how many molecules of the compounds which you would like to detect you have, and also if there is some, another molecules which may, may interference with the signal, from the molecules which you would like to detect. So yeah, I was always suggesting them, okay, So what you want to detect? Give me a sample. Tell me a little bit more, what is the concentration of the compounds what you want to detect. So what will be the range? Also, the Raman is really good for the organic, so more complex compounds, because there's not really too many other tools. I mean, the non-invasive tools. Yeah, otherwise you have to, take a sample, concentrate the sample, blah blah. So what? Yeah, I think again into this food industry. For sure, Raman will help, and I'm also sure there are some now commercial equipments, which they are using for that application. Uh, but the question is, what you want to detect and what is the concentration, how many molecules you have for the system, and how you will then design the system to get enough information. Sugar contents easy, acid, like you can measure from the apple, lemon, easy, but if you want to detect very small amount of some molecules, you have to check if the peaks and the signal is not covered by some other signal which is already there, and if you have enough signal. That's it.
Thanks very much. Thank you.
Okay, Martin, perhaps you could stop sharing your screen. I think that would just make it a bit easier for us to see if anyone's asking
Okay.
any questions. So while we're doing that, are there any more questions from the floor? I can't see anything in the chat, and if anyone wants to ask a question, just feel free to unmic or put something in the chat. Okay, so I had a question then, Martin. So, obviously, if you look at more conventional image analysis in phenotyping, RGB, chlorophyll fluorescence, whatever, a lot of the applications there are kind of based on populations, right? So with these more specialist technologies, are there throughput limitations, and do you see ways in which those applications may become more high throughput in the future?
So, okay, but what is I don't understand what is exactly the question. So
So the question I'm asking, right? So if you look at, standard phenotyping, a lot of the applications are looking at populations, and I'm wondering whether the applications you've spoken about today, the Raman, the X-ray and the neutron imaging, are they amenable to high throughput applications, and if not, what are the barriers to them?
Okay, so you're right. Yeah, this is the question, when we always talk about a high throughput system. Yeah, you're right. RGB takes milliseconds, uh, and then you need some time for the movement to prepare the end of the sample. X-ray, and this is what I said in the beginning. If you will scan the whole plant, it really takes minutes. And I mean, in the minutes, if I would like to have the 3D image of the plants, it will take 1 hour easily. Yeah, depends on the resolution, because our X-ray may have 50 micrometer resolution, and so that is the limit. But on the other hand, so what we developed, we developed the method to select object of interest, and then the scanner may just scan, and it's just 2D scan, may scan just the area, where is your interest, and then it's about again the seconds. It's not about it's not about milliseconds, but about the seconds for the X-ray. Neutron imaging. Once again, the problem is the neutron imaging is in principle at the moment the regulation and the source of the neutrons, but it's imaging. It's like RGB, so it's in the milliseconds. It's easy. Depends on how powerful is your source. Uh, and Raman for some components, it's again about the seconds, So, but it takes time, to point, to put your sensor closer to the. But then the measurement is second. for some components, I really need to have as the minimum like 1 minute to acquire this extremely low signal. So it depends what you want to do, but still, if I compare what other do, if you will spend like 2 minutes with the one sample, it's reasonable because you may do 30 samples per hour, and yeah, it's not, you will scan thousands plants, per day, but you still will have possibility to scan, 100 samples per day, which is still, okay. But yeah, you need, quite high number if you want to do good statistic and etc.
Okay, so thanks for that, Martin. Any more questions from the floor? No, I think that's everything. So again, I'd just like to thank you for that really interesting presentation this afternoon, Martin. And hopefully, if anyone wishes to join the next set of APGC presentations, Donna will keep those invitations coming out. So thank you again, Martin. Hopefully, we'll speak soon.
Thanks all.
Thank you.
Take
Bye.
care. Take care.